Thanks for coming by to view this poster presentation. My name is Taylor Barr, and I'm a second-year medical student at the West Virginia School of Osteopathic Medicine, and I worked on this project with Dr. Crystal Boudreau, who is an assistant professor of microbiology at the West Virginia School of Osteopathic Medicine in Lewisburg, West Virginia. So our project, we wanted to take a look at the novel coronavirus because it's pertinent, relevant, it's impacting everyone. There's no current definitive cure or prophylaxis. And we wanted to take a look and see what host cell proteins may help the virus replicate by direct interaction. And so it's been shown previously that the mTOR pathway has been implicated in the Middle Eastern respiratory coronavirus. So we want to take a look particularly at the mTOR pathway, at these proteins STK11IP, mTOR, LARP1, and CK2. And so we want to take a look at the protein sequence and structure, and taking a look at how the structure is interrelated with the function. And so identification of these areas could provide potential targets for antiviral therapy development. So you can go ahead and take a look at the slide for a quick overview, and we're going to go ahead and move on to the materials and methods now. All right, so first of all, I want to say that this is a theoretical analysis of protein sequence and structure. We used a couple parameters, and so you could use different parameters and potentially get different results. So how we worked through this was we got the sequences from Uniprot, then we put the sequences into NCBI Protein Blast, and you can see here on the bottom left here where we have that multiple sequence alignment view. And so we put the proteins in and sequences and we queried them, and we got just a list of different proteins that had similar sequences, and we filtered out the ones that were not homologs of that protein from different species. So we ended up just with the homologs. And so then we went through and looked at conservation position by position. Was every amino acid residue in that position the same, or if it was a conservative substitution? And then after that, we assigned each amino acid residue in every protein a charge designation, positive, negative, polar, and non-polar neutral. And then we use that to look at charge concentration in certain areas of the protein, and as well as concentration of particular amino acids in the protein. And so that's what we use our sequences of interest. We took those sequences and mapped them out onto the protein structure using UCSF Chimera molecular modeling software. And then also for STK11IP, there's not an elucidated structure for it, so we queried its sequence into the cath database, and it just returned a bunch of proteins with similar sequences or sequence motifs. Then we went through and selected a couple that had to do with viral immune response. And so one of them was the toll-like receptor 7 protein. And so we'll talk about that later. And then also we used JPRED4 as a secondary structure prediction algorithm to identify secondary structure for areas that don't have elucidated structures. Okay, so in the top left here, we have a couple of tables, or a few tables. A is just a breakdown of each protein conservation. So most conserved is mTOR at 92 or 95%, excuse me, and STK11IP is least conserved at 50%. Then in B, we have a breakdown of each protein by the amino acid charge residue charge designations. And then in C, we have a breakdown of each protein conservation by the designated charge residue designations. And then we have in the bottom left mTOR, and there's a few sequences here or domains called tetratrichopeptide repeat regions, TPRs, and so they're purported protein-protein interaction domains. And so there's 16 of them here in mTOR in this, and you see it here in the complex mTOR1 complex, and then TPR13 here is in pink, here in pink, and you see that it's the connection here between those two is unknown. Not elucidated anyways, structure-wise. And so this TPR13 is a protein interaction region, and it's open and available to interact with surrounding proteins. And it's interesting to note here that it does have a high concentration of charge, which makes it likely to interact with oppositely charged areas on different proteins. And then we have this interesting repeat or region here with alanine and threonine residues. Like I said, it's around this region, and it's not elucidated in the structure, so we predicted it with JPRED4. And there's no structure here, no red, no green to indicate helices or sheets. and you do see it's confident because of the high black bars here that it is unstructured. And then in the top right, we do have LARP1. And so we have this region of LARP1 here that is known to interact with 7'-methylguanosine mRNA caps. And opposite the side that interacts with the mRNA here, we see these 3 coils, which are highly charged. You can see it here in the table to the right. Each coil is significantly more charged than the full-length protein overall is. And so it's interesting to note that in a protein that interacts with mRNA, that a virus could potentially hijack, there is this area that it makes it available to interact with other proteins. In the bottom right, we do have the STK11IP compositionally biased region, and so that's just an area of protein with a lot of one residue in an area, and that's glutamate here. So the first half is not conserved, and so you can see here it's predicted to be non-structured. The second half is conserved and it has some structure to it that's predicted, but because it's unstructured in the first part, it could be a fold upon binding domain, which would indicate that it binds to a different protein here at this region, and it could cause the protein, the STK11IP, to fold and have a potentially new function. All right, and so just real quick here, we have on the left here, CK2, there's a region in the pink and purple up there on the left that is open, highly charged, available to interact with other proteins. And then on the right, we have STK11IP sequence mapped out onto the toll-like receptor 7 structure, potentially showing that it could form the same structure and a similar function as well. So in conclusion, these areas that we have isolated could potentially serve to target development for antivirals. And then also there's no structure elucidated for STK11IP. So in the future, it would be good to see that structure get elucidated. Thanks so much for watching the presentation.